Welcome guys to my YouTube channel Rare Science Simple. I am currently giving lectures on microscopy, and today I am going to cover the topic of confocal microscopy. So before moving forward, we need to understand why the confocal microscope was discovered, because fluorescence microscope was already available at that time. So the reason was this: out of focus fluorescence. Fluorescence microscope is also known as the wide field microscope and epifluorescence microscope, and it was having the drawback of out of focus fluorescence. So what is this out of focus fluorescence? Suppose this is your lens and light is coming from your sample, and this is the principal axis of the lens, and this is the focal plane. Okay, so when the light is hitting this particular lens and producing the image as the focal plane, this is known as the on focal fluorescence or on focal image. Anything above or below is considered as the out of focal plane. So for example, anything above this focal plane or anything below this focal plane is considered as the out of focus image. Means the image is not focused and it is going to be blurred image. This is known as the out of focus image because this is not being produced at the focal plane. So every sample basically has the focal plane. Suppose this is your lens and this is your specimen and this is your focal plane. So this specimen has a specific type of the focal plane, which is this one. Let's say this is the focal plane, and now light is coming here, here and here and here. So the image which is going to produced here, this is the on focal image, this image, and anything above and below is the out of the focal image. This is known as the out of focus image, and this image is going to be blurred, and this is the on focal image because this is going to be produced at the focal plane of the specimen. Okay, so now moving forward, what was the principle of the fluorescence microscope? So in the fluorescence microscope, we are using the light source. So light source will emit only the white light. So white light will hit the excitation filter. In the fluorescence microscope, excitation and the emission filters are basically chosen on the basis of fluorophores which you are using to label your sample. Suppose you have the sample and you are labeling suppose any organelle or any molecule with the FITC fluorophore. So FITC fluorophore has a specific kind of the excitation wavelength and the emission wavelength. So for example, it can have the excitation wavelength of 495 and emission wavelength of 519. So we will use the excitation filter here, which will only allow to pass the light of this wavelength. We will use the emission filter here, which will only allow to pass the light of this wavelength. So on the basis of fluorophore molecule which you are using to label your sample, you are going to use the specific type of excitation and the emission filter, and that you will set when you will perform the experiment in the fluorescence microscope, that you can set basically. Now we will use the diachronic mirror. What is the characteristic feature of diachronic mirror? Number one is this: in the fluorescence microscope, we are using the diachronic mirror. Number one thing is this: it is always set at the 45-degree angle. Number two, it is transmitting the light of one wavelength. which is the shorter wavelength, and emitting the light of larger wavelength. So two kind of light, it can either transmit the light or it can also emit the light. Not or, basically, and it can transmit the light as well as it can emit the light of the another wavelength, which is of larger wavelength. So now coming back to here, now this diachronic mirror, this particular light, if suppose we are taking the example of FITC, this particular light will hit this diachronic mirror and then it will go to the objective lens. Now this objective lens will go to the from this objective lens, this light will hit this particular specimen. Now as I told earlier, every specimen has the focal plane. Let's say this is the focal plane of this particular specimen. Okay, this is the focal plane. So what happened from this light will come like this and go like this and go similarly come like this and go like this. So the image that is going to produced when the light is hitting the focal plane of the specimen is the on focal image, but whatever is here and here is considered as the out of focal image. Let me clarify it here. For example, this is your objective lens and this is your specimen and this is your focal plane. This is your focal plane. So light will hit like this in case of the fluorescence microscope I am talking about, and like this. So therefore it is known as the wide field. The field is wide, and any fluorophore molecule which is present here and here and here and here, they are going to make the out of focal image, because this is not fixed. This is the field is very wide here. This is going to produce the out of focal image. You can say the illumination is not very fixed type, and therefore the light is hitting on the wide field of the specimen, and therefore the out of focal image is going to be produced by the fluorophore molecule which are not present on the focal plane. So this was the drawback of this particular fluorescence microscope, and as you can see here, this is the white field. So these are the fluorophore molecules. So the image that is going to be produced by these fluorophore molecule is going to be blurred, because these fluorophore molecules are present not on the focal plane of the specimen, but they are present out of the focal plane. So this was the drawback of this fluorescence microscope. Now what will happen? The light will be emitted, that will be of larger wavelength or lower energy, and then there will be the emission filter, and from the emission filter, that is going to be detected by the light. That is going to be detected by the detector, which could be the photomultiplier tube as well. Okay, so this was the principle of fluorescence microscope. Now coming to the confocal microscope. So in case of the confocal microscope, what the scientists did, they want to keep the illumination fixed. So majority of the fluorophore molecules are present only on the focal plane. The light is hitting only on the focal plane instead of out of focal plane. Okay. So for example, here you can see we are using, in this they basically use the laser light. Laser light will go to the pinhole, and there will be the excitation filter here. Okay, so there will be the excitation filter here. Okay, and light will, now this illumination is going to be fixed. It's not fixed, you can say focused. Illumination is going to be focused here. Illumination is focused because of this pinhole. Okay, now it will hit the excitation filter. This is your excitation filter. Again, that will hit your diachronic mirror, from the diachronic mirror to the objective lens, and then to the focal plane of the sample. So what you can see here, basically, if suppose this is your lens and this is your specimen, okay, and this is the focal plane of this specimen, basically, because every specimen is going to have the focal plane. Now the light, because it is more focused because of the presence of pinhole, it will be like this. Okay, so the field is now going to be reduced. You can see this in case of the fluorescence microscope. What was happening? This was the lens, this was your sample, and the field was very wide here. Okay, so here you can see many fluorophore molecules which are out of the plane. This is the fluorescence microscope, and this is the confocal microscope. And here you can see a smaller number of the fluorophore molecules, that is going to be the out of focal plane. But it will be there. It is not 100% perfect. Now to prevent the blurriness of the image because of this out of focal fluorophore molecule, they have used another kind of filter, which is, you can see here, this is the another kind of pinhole they have used. So whatever the light is coming from the out of focal plane is going to be stopped by this pinhole, and only the light which is present on the focal plane of this particular image is going to be produce the image via the photo detector. So there are two things they have added. One is the pinhole they have added in between the laser source light and the excitation filter, and the another pinhole they have added in between the diachronic mirror and the emission filter. So there will be one emission filter as well. So let me just draw that also. So here will be the emission filter. This is going to be the emission filter. So this is your out of focal plane. And this is your on focal plane, because you can see it is hitting the focal plane of the specimen. Okay, and this out of focus light rays is going to be stopped by this pinhole, and on focal image will not be stopped by this pinhole, and they will produce the image. Okay, so this was the principle of the confocal microscope. Now, why the laser light was used in the fluorescence in the confocal microscope? Number one, laser light is more stable. Number two, it generates less heat in comparison to white light. Number three, it has the coherence properties. Coherence properties means light rays are in the same phase. What does it mean? Basically, the light rays are in the same phase, same amplitude, same wavelength, and frequency. This is the characteristic features of the laser light, and therefore it was used in the confocal microscope. Then what should be the size of pinhole? Size of the pinhole you can decide it by this is the resolution power, you know the resolution basically. So 2 multiply by 0.61 lambda divided by numerical aperture, and you multiply it by the magnification. If you are using the 10x or 100x, so you can multiply it with the magnification. That will give you the ARRI unit, which is also known as the AU. So now you know the resolution. and resolving power. They are inversely proportional. Okay. So we want resolving power to be high for the microscope. In microscope, this should be high and this should be low. Okay. What is the resolution means? The distance between the two points should be less, but the resolving power is going to be high. So this is the resolution. So you can see if the wavelength of the light is less and numerical aperture is more, then you will be having the less resolution and more resolving power. So questions come sometimes on this also. So therefore I am explaining it here. Okay. So don't be confused by the resolution and the resolving power. They are inversely proportional. Now, the microscope, the confocal microscope is basically of two types. Okay. One is the laser scanning microscope. In this, we are using the scanner. This is the scanner. Okay. This is the scanner. So what happened? This is a scanner. Okay. So a scanner will scan your sample. Suppose this is your scanner which you are using. This will scan the light. Okay. And then there is an objective lens, and then there is your specimen. So it will divide your specimen of to the multiple slices like this. Suppose these are 1 slice, 2 slice, 3 slice, 4 slice. So first it will scan this particular slice, then it will scan this particular slice, then it will scan this particular slice, and then it will scan this particular slice and produce the image, and all the image it will superimpose to each other. All the images, what it will do, it will superimpose with each other, and then it will produce the tertiary structure of the image, which is also known as the Z-stack. This will produce the tertiary structure of the image. Okay. Tertiary image, you will 3-dimensional, you can say 3-dimensional image you will be getting, which is also known as the Z-stack. Okay. So this is the basic of laser scanning microscope. In this we are using the scanner, and via this scanner, this particular specimen, because this is your specimen, let's say. So everything, every single slice is going to be scanned by this particular scanner, and then it will produce the image of that slice first, then to this slice, then to this slice, and because of that it will superimpose all the images, and then it will produce the 3-dimensional image of your sample. Okay, so this is the advantage of this, and everything else is going to be same in the this microscope. You can see the laser light, you can see the diachronic mirror, you can see the pinhole, photo multiplier tube, pinhole here. There will be the excitation and emission filter also, which they have not mentioned here, but it will be here. So suppose this is pinhole, so here will be the excitation filter, and if this is pinhole, there will be the emission filter. Now, what are the drawbacks of this particular laser scanning microscope? One is the phototoxicity. So phototoxicity means because of the speed, it is scanning the many slices of your specimen, and therefore it is taking more time. So speed is very less in this case. So what happened here? Speed of the image production is less. So you need to sit in front of the microscope for the longer time, and therefore your sample is going to be exposed to that particular light for a more time, and therefore it will produce the reactive oxygen species, and that can destroy your DNA or protein of your sample. So this is known as the phototoxicity of the sample. This photon light which are by which your sample is exposed for the longer duration of time, it can cause toxicity to your sample by generating the reactive oxygen species, which will degrade your DNA or protein. The next is photo bleaching. destruction of the fluorophore. You are exposing your fluorophore molecule for the longer duration of time, and because of the photochemicals, there will be the destruction of fluorophore. So you can write here, this is the destruction of fluorophore because of the photochemical. Destruction of fluorophores because of the photochemicals. Then depth penetration. If your sample is very thick, then you need to use the multi-photon laser light, and less axial resolution, and the uses of different mounting media for live and dead cells. Suppose you are using the live cells and your mounting media is sensitive for the pH, then your live cells will respond to that media differentially once the pH changes. So different mounting media you need to use. In case of the live cells, you need to use the media which is pH insensitive. Okay, so like this, these are the drawbacks. And what is the axial resolution? One is the lateral resolution and one is the axial resolution. If suppose these are your 2 points in your image and light is coming perpendicular to this, then this is known as the lateral resolution, and axial resolution is this one. If these are your sample, this is your axis, light is coming parallel to this. This is known as the axial resolution. So this is the image is going to be produced like this here, and here it is going to be produced like this here. Okay, so this is your light. That is coming perpendicular, and here it is light which is coming parallel to the axis of your samples. So axial resolution in case of the confocal microscope is less, because your samples, if you think about, your sample will have like these many points. Okay, so for this it is the lateral resolution, and for this is going to be the axial resolution. So that's how you need to think. This is the lateral and this is the axial resolution. Now another kind of microscope is the spinning disc microscope. In this microscope, basically you are using the micro lens, and you are using the two kind of disc here. One is this disc, and another one is two disc you are going to use. So this disc has multiple holes. It's like 1,000 holes in a disc. So at a time you can scan your sample 1,000 times. So the speed is going to be very fast here. Okay, so light will come from this, pass through this hole to this hole to this hole, and it will hit a multiple points in your specimen, and therefore the speed is going to be high in this particular kind of microscope. Rest all is going to be same. You can see the laser light you will be using, laser illumination. There will be the micro lens that will create the more focused light, then there will be the diachronic mirror, and then you are using the spinning disc here, and then it is hitting the objective lens, then to the sample, and rest of the things are going to be same only. So in this particular microscope, first of all, we are not using any photo multiplier tube. We are using the CCD camera. Why? Because the CCD camera has the high photon efficiency, higher quantum efficiency. What is the quantum efficiency? If suppose 100 photons are hitting, let's say if suppose the quantum efficiency of the photo multiplier tube is 30%, it means What does it mean? 100 photons are converting into the 30 electrons. In case of the CCD camera, the quantum efficiency is higher than the 30%. Let's say if it is like 50% or 80%. So what does it mean? Let's say if this is your CCD camera and it has the quantum efficiency of 80%, it means 100 photons, if they are hitting 80 electrons, they are converting into the 80 electrons. So the quantum efficiency is high, so image is going to be more, you will get a very better, or you can say the contrast image you will get. Okay. But there are some drawbacks of this particular spinning disk microscope. So number one drawback, number one is this: the disk alignment. You can see light is here. You can see what does it mean if we are talking about disk alignment. The light is passing through this hole, then it should emit through this hole, and it should exactly pass to the this particular second disk at the same position. So there should be the disk alignment. This light should not pass all the way, out of this plane. So this alignment is very important. You can see this is light is coming from here, then coming here, and then the emitting light is again passing through this hole, and then the hole that is in the alignment of this particular disk, it should pass through that particular hole only. So this alignment is the major drawback of this particular scanning disk microscope. Then number two is disk alignment, the movement of the disk and the movement of disk with camera. It's like this: if suppose you are roaming here and there and I am fixing the camera at one position, then I cannot take your picture if you are moving. So the camera movement and the disk movement, that should also be aligned. Suppose I am taking your picture and you are moving, I cannot take your picture because you are moving. I need to move also, then I can take your picture. So there should be the movement of the alignment of the movement of the disk with the camera movement. This is the second drawback. And third one is the crosstalk. Okay. So I don't know if I have any here. We can, okay, so I can just erase this basically now. What is the crosstalk? What is the crosstalk? If suppose this is your sample, okay, and light is coming from this particular hole, okay, and if suppose this particular hole is very near to this hole, then this light can also pass through this hole, and this is known as the crosstalk. This is the another problem with the spinning disk microscope. So let's say if these are the two dots, you can see, then the transmittance which you can calculate is going to be d by s square. D is basically the diameter of this particular hole, and s is the basically space between these 2 points. Okay, so lesser the transmittance, more the resolving power. Lesser the transmittance, more the resolving power. Okay, so for this, what you need to do, you need to keep the diameter less. Suppose you are taking the high diameter, let's say, and the speed is also, and the space is also less. These are having high diameter and speed is less. So light can pass through this hole, but it can also pass through this hole. The chances are very high. But suppose if it is having diameter is less and the space is also high, so the light which is passing here, the chances of passing light through this hole is going to be less. Okay, so the transmittance is going to be less and therefore the resolving power is going to be high. So that's how you can also calculate the this is the formula which you can remember here. So coming back to this particular spinning disc microscope, the major advantages of light and the drawbacks, as you know, the alignment, crosstalk, and movement, alignment between the disc movement and the camera movement. If these discs are moving, camera should also move. And transmittance, if you want resolving power to be high, so the whole distance is going to be high, should be high, and the diameter of these holes should be less. Then there will be less transmittance and more resolving power. So with this note, we can do one question which has come in CSIR, and we will do many questions at the end of my lectures when I will finish the series of microscopy. But let's now do this particular question about the laser scanning confocal microscope and try to give the answer by yourself. So laser scanning confocal microscope is a wide field technique. It's not a wide field microscope. Fluorescence microscope is the wide field microscope. So this is the wrong statement. Kohler illumination system is the light microscope basically. So Kohler is what? Is the Kohler is the light microscope. It's the light microscope. This is the wrong statement. Now the second statement, which is like this: spatial resolution is higher than that achieved in the wide field imaging. Definitely it is higher and could be obtained if only the central portion of the area disk is used to form the image. If it is central portion, it is more focused, if the pinhole is more focused. So this is the correct statement. Then scanning mirror sweep the excitation beam over the sample point to build the three-dimensional image. This is again correct. Now fourth one is an altered pinhole size does not make any impact on the resolution. It makes the impact. You know, the pinhole size is going to be decided by lambda Na 2 multiplied by magnification. So whatever the pinhole size, it will definitely decide your resolving resolution and the resolving power. So this is going to be the wrong statement. A photomultiplier, a tube in LSCM helps in generating real color of the fluorophore? No, it is not. So the correct answer is this one and this one. The real image is going to be produced by the fluorophores, the wavelength of the light they are emitting or they are exciting. On the basis of that, the color of the image is going to be produced. So not the real color of the fluorophores. So this is the correct statement. B and C is the correct statement. So with this note, I am finishing my lecture, and next lecture I will cover the topic of phase contrast microscopy. And if you like the content of my channel, kindly subscribe my channel. Thank you so much.